Hello everyone, in this lecture we are going to discuss about botulism. The botulism is a condition caused by potent protein neurotoxins elaborated by Clostridium botulinum. It is characterized by symmetrical descending flaccid paralysis of motor nerves and automatic nerves, usually beginning with cranial nerves, which are present in our brain. So this Clostridium botulinum is an anaerobic gram-positive organism that can form spores. And these spores are ubiquitous in nature, which means they are resistant to heat, light, drying, and radiation. Spores survive even boiling after 100 degrees Celsius. And there are some specific conditions for the germination of these spores. One is anaerobic conditions, warm conditions between 10 to 50 degrees Celsius, and mild alkalinity, which enable the spores to germinate and cause botulism. And there are some neurotoxins which are secreted by Clostridium botulinum that cause botulism in different kinds of organisms. So there are different kinds of neurotoxins secreted. Neurotoxin A, B, C, D, E, F, and G. So neurotoxin A and B causes botulism in humans, and B and C causes botulism in horses. B, C, and D in case of cattle, and C in case of sheep. In case of dogs, it is C and D. Avians, C and E. Mink and ferret organisms, A, D, and E. So these are the following neurotoxins causes botulism. And epidemiology. This botulism was first emerged in the Republic of Georgia, Armenia, where illness is associated with risky home canning practices. And in the mid period of 1990 to 2000, median number of foodborne cases are noted, and the mortality rate was 25% prior to 1960. There are different kinds of botulisms. One is foodborne botulism caused by these neurotoxins, and intestinal botulism caused through spores which further secrete the neurotoxins, and wound botulism, though these spores which are secreted by Clostridium will germinate, infect the wound and germinate inside the wound and spreads enters into the body. And adult botulism is by ingestion of perforated toxins. And infant botulism was characterized by constipation, food feeding, sucking, and swallowing. Pathogenesis. So the neurotoxins, the spores infects the wounds and germinate over there, enters into the body, enters into the bloodstream, binds to cholinergic nerve endings, and further inhibits the release of exciting neurotransmitters like acetylcholine. So this spore or inhibition of secretion of neurotransmitters causes flaccid paralysis, and also followed by certain serious conditions like double vision, dysphagia, difficulty in breathing, speaking, and respiratory failure. Mechanism. There is another kind of mechanism where the neurotoxins will be absorbed from the gut through foodborne diseases, foodborne toxins, and they binds to the nerves of peripheral nervous system and cranial nerves, followed by proteolysis by the light chain of botulism toxin. So they attack the snare proteins in the neurons. So by affecting the snare proteins, inhibition of release of acetylcholine at the synapse causes lack of muscle contraction, finally leads to paralysis. Diagnosis can be done through toxin neutralization and mouse biopsy. where the collection of samples like serum, stool, and suspect food is diagnosed in order to measure the concentration of the toxins. And infant botulism, characterized by serum, vomit, gastric aspirate, and suspect food. Through mouse bioassay, detect zero point 0.3 nanograms of toxin in the sample. Through cultures, the fecal and gastric specimen cultures, anaerobically results will be appeared after 7 to 10 days. So there are some unique features to help in diagnosis, where disproportionate cranial nerve palsies, more hyptonia in facial muscles than below neck, lack of sensory changes. So these are some features which help in the diagnosis. For the prevention, as it is a natural disease, so there are some natural techniques like whenever we bring the canned food to home, boil the canned food for at least 10 minutes, and the appropriate immediate medical care must be taken for the wounds. Avoid injectable street drugs. So vaccination is provided for botulism, where botulinum pentavalent toxide. So it is actually in use for laboratory, which who are working in laboratory workers and military persons, and it provides long lasting immunity. So this is the brief introduction of botulism. Thank you.